This was my high school yearbook quote. I guess you can tell, yep, I'm a nerd. And I've spent most of my life in a laboratory, about 80% of it. And what I'm here to talk about is I'd like to encourage you to spend some time in a lab too. This is the age of biotechnology. Last century was the century of physics. This is definitely the century of biotech. And whether you know it or not, it's touching almost every aspect of your life. There are companies out there that are hacking your food. They're hacking the materials of your daily life, rubber, plastic, even fuel. And they're also hacking the human body. Personal genomics, even genome editing is on the horizon right now. So my contention is that a working knowledge of genetic engineering, just like you have a working knowledge of electricity, is an essential skill for the 21st century. Now, what is biohacking? Well, this is kind of what it looks like, or what it looked like back in 2009 when I founded Genspace with some other friends. And it was essentially people trying to do biotechnology experiments in an unconventional environment. People that didn't necessarily go through the rigorous training or whatever. And what we did was kind of simple at the time. The movement was called DIY Bio. It was kind of a coming together of several factors. The maker movement, where everyone wants to make things. Synthetic biology, which is a discipline that makes biology easier to engineer. And also the fact that reading and writing DNA code is getting ridiculously cheap. You can actually buy a bacterial gene for about $150 right now. So that was what fueled this movement. And so eventually it coalesced. There are probably thousands of people right now in the world doing this, and most of them have developed what we call community laboratories. So what a community lab is, this is Genspace. This is the one that I direct right now. And it's a space where you have shared equipment, shared supplies, and it acts as a platform for innovation, a platform for exploration. It's open to the general public. It's kind of like a gym. You pay a membership fee. You get some safety training, some mentorship. We run classes. We have lectures. It's a very open and vital meeting place for people. And you can explore anything you want. As long as it falls within certain safety guidelines, you can explore something that has been done 1,000 times before, but it's new to you. You can do something that's really innovative, or you can do nothing but talk to other people about what you'd like to be doing. So what kind of projects happen there? Well, this is a game that actually is played with live paramecium and an iPhone. It's kind of a little video game. We've demoed it at a street fair not long ago. We also have group projects at Genspace, what we call our citizen science component open to the general public. This one happens to be barcoding plants from interior Alaska. Every year I go to Alaska, I bring back some samples. It's an ecology, environmental project, and we get everybody involved. But you may be asking yourself, well, you know, this is fine if you're kind of into nerdy science. What really does this have to do with my daily life? Well, I put it to you. Do you want to know what's in your food? Do you care? Amazingly enough, I just judged a high school competition in what's called DNA barcoding, the same thing we use to identify plants in Alaska. It's very useful for identifying whether or not what's on the label is on your table. And about 50% of the samples that the students brought back were not labeled properly. So, you know, the FDA, they try, but they're horrendously understaffed. They can't be everywhere. And isn't it nice to be empowered to take this into your own hands? This is what this century is going to be about. With a little bit of training, you can do this very easily. For example, when they had that horse meat scandal in the hamburger in Europe, I think it was last year or the year before, people flocked to these DIY labs, and they could test their samples for themselves. Also, personal genomics is a big deal. Now, this is a very interesting project. This is an artist named Heather Dewey-Hagborg who worked in our lab. She was curious, how much can you find out about a person from something they leave behind? So she collected cigarette butts, chewing gum, hairs off the street, and she was able to analyze them to the point where she had enough information to print a 3D face of a person she'd never met. Now, of course, this technology isn't 100% accurate yet, but you can bet that law enforcement is putting a lot of money into this, and it's only going to get more and more accurate. So what else can you tell about a person from that? Well, she could potentially know more about you than your doctor. So why not learn this for yourself? Why not explore your own genome? This was us at a conference. We set up a table with some relatively simple equipment. I don't know if you can see, but there's actually a tea kettle on the table. A very interesting piece of scientific equipment. And so we had people swab their own cheek cells, and some of the work we ended up doing in the hotel room. I mean, it's that easy. And everybody went home with a little glimpse into a part of their genome. This happens to be a gene called CCR5 that is one of the major receptors for the AIDS virus, and about 1% people are double mutants that have a deletion in this gene, and they're essentially not susceptible to the primary form of AIDS. So, an interesting thing. This is a project where a bunch of us decided to do a hackathon in collaboration with Genomicon and with Synbiota. We put together a pathway that makes a particular drug. And there was a question as to whether you could put this enzymatic pathway together in different ways. And this was facilitated by this really cool robot called the OpenTron, which is an automatic pipetting robot that was actually made by a guy who's a Genspace member and some of his friends, completely open source. And so we had a whole hackathon making different incarnations of this pathway in bacteria, which was kind of cool. I mean, that's actual biomedical research being done in an unconventional space. So we work with a lot of different organisms at Genspace. You can work with all sorts of bacteria which are not pathogenic to humans. This is the Hello World experiment where you turn them fluorescent green or fluorescent red. We also work with plants, with fungi, just about anything that's not a vertebrate animal we can work with. Even mouse cells. So this is also a project that is, it's representative of what sort of thing that you can do with those cells. It's a biomaterials project. This is bacterial cellulose on this little model chair for the seat and the back. It's a product not of trees, but of bacteria made out of a bacterial culture. And it was a project of some students, much like these students, that were part of a genetic and engineering team. And that's another really good use for these spaces, is there isn't enough lab space and there isn't enough mentorship for this incredible technology that's going to essentially take over our lives. And yet it's taught in schools, but not to the extent that it's going to be needed. So we love it when teachers come to Genspace and use the facilities with their students. We love it when high school students come and say they want to do a project. These kids are now doing a genetic engineering project as part of a competition in our lab. And I think if you need another reason why biotech should be accessible to everyone and also something that people should be literate in, I think it's this, that there's a lot of stuff coming down the pike here. There's a lot of stuff that has social and moral and ethical implications. And I don't want the decisions made about this technology to be knee-jerk. I want it to be made from the point of understanding, not from fear or ignorance. And it's really hard to be afraid of something that you've done side by side with your kid in our lab. So that's another good reason to do this. This is our latest project. It's called Open Plasmid. And what we're hoping that it is an open platform that's going to do for biohacking what the Arduino did for electronic hacking. I think I want to end with this note that this stuff is not only fun, but I think it's crucial. And I really encourage people, get to know this stuff. Really get your hands dirty with this stuff. It's not scary. It's really critical. And hey, you never know what great things you can bring to life, right? Thank you.